My dear students, for 12th zoology, biotechnology and its applications, we are dealing with the definitions, principles, processes of biotechnology. It is the technique of using live organisms or enzymes from organisms to produce products and processes useful to humans. In this sense, making curd, bread, or wine. These are old ages practices: curd, butter, and wine. Curd, bread, and wine. We can see also, apart from the biotechnology subject, these are also the products of biotechnology, which are all microbe-mediated processes. These are microbe-mediated processes. Biotechnology is a very new subject introduced in 1970, but so far these techniques are concerned, these are very age-old techniques. They were also microbe-mediated. We can speak that these also form the core fields of biotechnology. The principles of biotechnology include there are some core techniques that enabled birth of modern biotechnology. Today, it is used in restricted sense today to refer to such of those processes which use genetically modified organisms to achieve the same on a larger scale. Further, many other processes techniques are also included under biotechnology. For example, in vitro fertilization lead to the formation of the test tube babies. Synthesizing a gene and using it, developing a DNA vaccine, or correcting a defective gene are all part of biotechnology. The two core techniques that enabled birth of modern biotechnology are: number one is the genetic engineering. This is one of the core branches or core techniques that give to the birth of modern biotechnology. It is a technique to alter the chemistry of genetic material. That genetic material is DNA, deoxyribose nucleic acid. To manipulate this DNA, or we can say to alter the chemistry, of DNA by the techniques of molecular biology, to alter the chemistry of DNA to introduce these into host organisms and thus change the phenotype of the host organism. When this DNA can be studied by the molecular technology, it can be cut by the specific molecular scissors into pieces. A useful piece can be engineered or fabricated into the host cell to produce a desired trait. That desired trait would be very fruitful. That desired trait would be advantageous and very productive. That desired trait is incorporated in the host organism, thus to change the phenotype of the host organism. This technique involves genetic engineering, cutting DNA by molecular scissors, introducing this desired piece of DNA into a host cell to produce its desired effect, involves the gene manipulation, or what we call as genetic engineering. The second most important principle is maintenance of sterile microbial contamination-free ambience in chemical engineering processes to enable growth of only the desired microbe or eukaryotic cell in large quantities for the manufacture of biotechnological products like antibodies, antibiotics, vaccines, and enzymes. This is the second important principle that gave birth to the modern biotechnology. We should understand the conceptual development of the principles, how these principles got developed. This is necessary to study the conceptual development. You probably appreciate the advantages of sexual reproduction over asexual reproduction. The former provides opportunities for variations in formulation of unique combinations of genetic setup, some of which may be beneficial to the organism as well as the population. Asexual reproduction preserves the genetic information, while sexual reproduction permits variation. So the sexual reproduction is more advantageous than the asexual one because this conserves the gene store, and the sexual type of reproduction forms the basis of variation, that is due to the variety of combinations of different genes from the different types of populations into the one organism. This sexual reproduction involves the crossing over. It involves the meiotic cell divisions, by which the variations get created due to multiple combination of genes, varied combination of genes. Asexual reproduction preserves the genetic information, while the sexual reproduction permits variation. Traditional hybridization procedures used in plant and animal breeding very often lead to inclusion and multiplication of undesirable genes along with the desired genes. The techniques of genetic engineering include creation of recombinant DNA. This genetic engineering produces recombinant DNA. A new DNA is formed by incorporating a beneficial or advantageous segment taken from another cell and making a new genotype. Most likely, use of gene cloning and gene transfer. It involves gene cloning and gene transfer. Gene cloning is the technique of providing desired variety of gene segments, and gene transfer is the transfer of genes by the genetic engineering from one organism to the another organism. This overcomes this limitation and allows us to isolate and introduce only one or a set of desirable genes without introducing undesirable genes into the target organism. This is the development of principles of genetic engineering that form the core technique in the biotechnology. This is all for today.